Meiosis, a single diploid cell with 46 chromosomes, is separated into 4 haploid gametes with 23 chromosomes. Homologous chromosomes are chromosomes that share the same genes at the same loci positions and share the same structural features such as size, binding pattern, and centromere position.
Get excited. Here we go.
Meiosis can be divided into 2 parts, meiosis I and meiosis II. Before meiosis begins, DNA replication takes place during the S phase of the interphase. The replication of chromosomes creates sister chromatids which are attached to each other at the centromere. In prophase I, the homologous chromosomes undergo a process called synapsis, in which they pair up to form bivalents. The non-sister chromatids cross over during this phase. This results in recombination of alleles and is also a source of genetic variation in gametes. Chiasma is formed when neighboring non-sister chromatids are cut at the same point. The junction is formed when the cut sections attach to the open end of the opposite non-sister chromatid. As a result, alleles are swapped between non-sister chromatids. This is called recombination. In metaphase I, the bivalents line up along the equator. Random orientation can occur, and this is a significant source of genetic variation. There are around 8.38 million possible orientations in metaphase I and II. In anaphase I, spindle fibers contract, and the homologous pairs are separated and pulled to opposite poles. In telophase I, new nuclei are formed and the cytoplasm begins to divide by cytokinesis. The nuclei are no longer diploid. They each contain 1 pair of sister chromatids for each of the species' chromosomes. The cell will continue to grow during the interphase II, but there will be no synthesis phase. In prophase II, the nuclei break down. There will be no crossing over at this stage. In metaphase II, the pairs of sister chromatids line up along the equator. Spindle fibers are formed and attached to the sister chromatids at the centromeres. Random orientation here also contributes to variation in the gametes. In anaphase II, the spindle fibers contract and the centromeres are broken. The pairs of sister chromatids are pulled to opposite poles. In telophase II, new haploid nuclei are formed. Cytokinesis then splits the cell into 4 haploid gamete cells. Fertilization of these haploid gametes will produce a diploid zygote.
Non-disjunction refers to the chromosomes failing to separate correctly, resulting in gametes with 1 extra or 1 missing chromosome. Individuals with Down syndrome have 3 copies of chromosome 21. One of the parental gametes had 2 copies of chromosome 21 as a result of non-disjunction. The other parental gametes was normal and had a single copy of chromosome 21. When the 2 gametes fused during fertilization, the resulting zygote had 3 copies of chromosome 21. This is known as trisomy 21, which leads to Down syndrome. The law of independent assortment states that the separation of alleles for one gene will occur independently of the separation of alleles for another gene. The orientation of any homologous pair is random and does not affect the orientation of any other homologous pair. Any one of a pair of alleles on a chromosome has an equal chance of being paired with or separated from any one of a pair of alleles on another chromosome. This means the inheritance of 2 different traits will occur independently of each other.